Good morning, everybody. Today I'm going to be talking about fecal source tracking and land use associations for antibiotic resistance genes in private wells. I'm presenting to you today from Wisconsin. That's where our laboratory is. Antibiotic resistance is a growing public health problem. This is a situation where someone will get a bacterial infection. They can't fight it off on their own, and so they get treated with antibiotics, but those antibiotics are ineffective because the bacteria causing the infection are resistant to them. The use of antibiotics in human medicine and livestock production selects for resistance in bacterial populations. That resistance can take different forms, but whatever the mechanism, it's mediated at the genetic level by antibiotic resistance genes, and that's the contaminant I'll be talking about today. So these contaminants can make their way to the environment, and once there, we have two general concerns. One of them is that the environment is a potential point of exposure to these contaminants for humans and for livestock. And the other concern is a little more broad, that the environmental compartments can serve as reservoirs where the bacteria exchange genetic material and where potentially novel resistance can emerge. And thinking about water specifically as an environmental compartment, we do know that these antibiotic resistance genes can be found in groundwater and that they can originate in human wastewater and livestock manure. On the other hand, we don't know, though, the prevalence of these contaminants in groundwater, how common they are, and we don't know about the relative contribution of the various potential sources of them to groundwater pollution. And a way to illustrate these knowledge gaps is think about a typical study that's done in this field where researchers will identify a suspected source out on the landscape, like a wastewater treatment plant, and then they will go test the groundwater right nearby and to establish a link between the source and the contaminants in the groundwater. And so those studies are good for establishing those links, but that type of targeted sampling makes it difficult to know how common these contaminants are across the landscape more generally. And these studies often focus on a single potential source, which means it's a challenge to compare the contribution of the various sources of these contaminants. So those two knowledge gaps lead to the objectives for this study. We want to quantify the occurrence of antibiotic resistance genes in groundwater from a randomized selection of private wells. And number two, relate that contamination to human and bovine fecal contamination of the private wells. And then third, relate that contamination to various source and transport risk factors. This study took place in eastern Wisconsin, Kewaunee County. This is a rural agricultural area. There are septic systems spread throughout the county and plenty of dairy cattle. And therefore, there's manure and septic system effluent as potential sources of contamination on the landscape. And those sources are sitting on top of a vulnerable aquifer. It's vulnerable because it's made of fractured carbonate rock, and those fractures allow water and contaminants to move very quickly from the surface to the groundwater and to move through the groundwater quickly and with little attenuation. And we have documented contamination of private wells in this aquifer by both human wastewater and cow manure. So for this study, we're sampling private wells. We sampled wells over the course of a year to make sure we sampled during each of the 4 seasons. And then at our lab, we tested these private well water samples for antibiotic resistance genes and microbial source tracking markers. In microbial source tracking, we test samples from microorganisms that are specific to a certain host to identify sources of contamination. So for example, some bacteria only grow in the guts of humans, are only shed in human fecal material, and therefore when we detect it in groundwater, we know human wastewater was a contamination source. We had a total of 138 well water samples. We concentrate large volumes of well water to improve our detection limits. And then we analyze those samples by quantitative polymerase chain reaction. This is a genetic testing method. We were looking in this study for 5 antibiotic resistance genes and a related gene called the class 1 integron gene. All of these are associated with antibiotic resistance in some way, and all of them have been found in various types of manure and human wastewater. For identifying fecal sources, we tested for a variety of human and bovine associated microorganisms. I want to note that for this study, the genes in particular are the contaminant of interest. These genes, they can be carried by pathogens, but they can also be carried by non-pathogenic bacteria. Moving on to the results, the table shows the number of well water samples and the percent positive for each of the gene targets that we were looking for. And a couple of points here about our occurrence results. That first, we saw that all of the genes we looked for were detected in at least a couple of wells. And when you look across all those gene targets, 46% of the samples were positive for at least one. So these contaminants were not uncommon in the private well water. The third column of the table shows occurrence values from other studies in the literature for groundwater. And in general, the percentage of samples positive that we saw was on the lower end of ranges of what has been previously reported. There are a lot of potential explanations for why they might be. The one that I want to point out as a potential explanation relates to our study design and objective, and that is we were randomly selecting wells as opposed to many of these previous studies were sampling groundwater near sources that they suspected would be contributing these contaminants. The next thing I want to look at is the co-occurrence of these antibiotic resistance genes with the fecal contamination of the private wells. For this type of contamination to occur, the ARGs need to be in the fecal source, then that fecal source needs to make it to groundwater. And so I want to look at the co-occurrence in two ways. First, in terms of the probability of antibiotic resistance gene detection, given a fecal source was present. For example, of all the wells that were positive for human wastewater contamination, how many of them also had an antibiotic resistance gene? And then second, we'll look at the prevalence of antibiotic resistance genes by fecal source. For example, of all the ARG detections we saw, how many of them co-occurred with manure versus with wastewater? Before we get into the specific fecal source category results, though, there's an overall observation to make, which is that these contaminants co-occurred with both human and bovine fecal sources, and they were also detected in the absence of either fecal source, which is not unexpected. The table now is showing us the percentage of fecal positive samples that were also positive for antibiotic resistance genes. So the first column is the fecal source category that was detected in the sample. The second column is the number of samples positive for that fecal source. And then the remaining columns are the percentage of samples within that fecal source positive for each antibiotic resistance gene. So for example, starting at the top row, neither fecal source was detected in 69 of 138 samples. And of those 69 samples, 1% were positive for this ERM-B gene, et cetera, across the other gene targets. So if we step back and look at these results together, I think there's an interesting trend that emerges. When neither fecal source was detected in the private well water, the probability of detecting an antibiotic resistance gene was relatively low. When just human or just bovine fecal sources were contaminating the private wells, the probability of detecting the antibiotic resistance genes was moderate, relatively speaking. But then when both fecal sources were present in the sample, the probability of detecting these genes was generally higher. And that holds true, I think, for several of those gene targets. And it's also, biologically plausible in that if multiple fecal sources are influencing one well, there's a higher probability we're going to see these contaminants showing up. The second way I want to look at these data is to take away the percentages and just look at the raw counts of positive samples and focusing specifically on the comparison of human versus bovine, because that was one of our key objectives. And so we see human 25 samples were positive, bovine more were positive at 35. And then for each of the individual gene targets, there are a couple of more detections associated with bovine manure. And then when you get to the end of the column for total detections, we see that there were more ARG detections associated with manure than with wastewater. Now, on the previous slide, it looked like the probability of these contaminants being present was similar regardless of fecal source. But because more wells were positive for bovine manure, overall, more genes were associated with, more antibiotic resistance genes were associated with bovine contamination. And this is, the fact that we saw more wells positive for bovine manure is consistent with what we know about the landscape in Kewaunee County. There's about 2.5 times more manure out there on the landscape than there is effluent from septic systems. So bovine manure is simply a larger potential source. The next thing is I wanted to talk about factors that might be associated with these contaminants. We looked at 56 individual risk factors in these 6 general categories: agricultural land use, septic systems, well construction characteristics, rainfall geology, and groundwater recharge. We used multivariable modeling to examine associations between the gene targets and the risk factors. The box on the left lists those risk factors that were associated with at least one of our targets. And I wanted to show you an example of what these relationships can look like. So the plot on the right shows on the x-axis the number of septic systems within 3,000 feet of a well, increasing, going from left to right. And on the y-axis, it's the probability of detecting this TET-A gene. And the solid blue line is a positive trend showing that the more septic systems that were near the well, the higher the probability we had of detecting this specific gene. And the last thing I wanted to talk about, moving more into implications, is, what does this mean? Well, the fact is the role of water and environmental compartments in the broader public health issue is still undefined. It's something the field is working on. A way that we're approaching this is to think about exposure to antibiotic resistance genes through private well water. And we use a modeling technique called quantitative microbial risk assessment to assess that risk of exposure. And there are 2 points I want to make that we glean from this risk assessment. The first relates to what we already talked about, the contribution of fecal sources. When we do this risk assessment, we see that exposure to some antibiotic resistance genes is more likely when a fecal source is present than when absent. So for a specific example, when manure was present in private wells, there is about a 2 times greater risk of exposure to the TET-A gene than when neither fecal source was present. But when human wastewater was present in wells, the risk of exposure was not greater than when no fecal sources were present, comparing it to background levels, so to speak. The second point is stepping back a little more broadly and trying to place drinking water exposures in the broader context of other places people might be exposed to these contaminants. And I do have to say there's very limited data, very few studies available for these comparisons. So these are by no means definitive statements. It's more like tentative observations trying to illustrate where we want to go with this. And so I used a pictorial inequality here to show that based on our data in comparison to others, that exposures via drinking water are about as important as foodborne exposures, which are more important than exposures via recreational water. And so that's the kind of context we're trying to find for these environmental contaminants.
2 minutes remaining.
Thank you. The last thing I wanted to mention is that the research I just covered is available in 2 papers that came out in the last couple of years. And with that, I'm done, and we're happy to take any questions.
Okay, great. We could take one question if we have one. Well, maybe I'll ask one. Are the residents in the area that you studied aware of what's going on, and are they concerned,
Yeah,
their reaction?
definitely. And long before we did this study, groundwater quality was a big issue there. There's, it's a dairy heavy region. Private well owners are worried about their water quality from that perspective. And there's been an argument for a long time about the effects of agriculture on groundwater quality in terms of manure and other things. But, the dairy industry points to the fact that there's all these septic systems. So the first part of this study was actually that fecal source tracking to try to identify the relative contributions just to private well contamination in general. And we came back and added this. So groundwater quality is a big issue for them.